Hello students, this is Dr. Selvan, Associate Professor, Department of Periodontology, Vinayaka Mission Sankaracharya Dental College, Salem. Today we are going to see the topic on defense mechanism in gingiva. In this topic, we are going to cover the introduction, gingival crevicular fluid, saliva, leukocytes, and at last the overall summary. Now, coming on to the introduction, like our body, the gingiva is also exerted by various pressures and stimuli. So, in response to these stimuli and pressures, they will be giving the host responses. Now, let us see what are all the sources of these host responses. These can include the epithelial surface, immune responses, the saliva, and the leukocytes. Now, coming on to the gingival crevicular fluid in detail. Now, the contents, especially the cells, bacteria, and biological mediators, can be seen in the GCF. It was detected as early as in the 19th century. It was first elucidated by Weyrhag. Cross and Brill, they conducted various studies on this GCF, and they found that this GCF fluid, it is excited from the blood vessels into the connective tissues, which later on moves to the gingival sulcus. In addition to this, there are various potential markers that can be found in the GCF. These markers, these can be used to find whether the periodontal disease is in its active stage or in its inactive stage. It can also be used to find whether our human host body is returned to its homeostasis. Now, coming on to various methods of the GCF collection. There are various methods of GCF collection like the paper strip method, twisted threads, intraclavicular washings, and the microcapillary pipettes. Among the paper strip method, there are two mechanisms. The one is the extraclavicular, the second one is the intraclavicular method. Now let us see these methods in detail. Now coming on to the paper strip method, as I said earlier, it is of two types based on the placement of the paper strip. One is the intraclavicular method, the second is the extraclavicular method. In the intraclavicular method, the absorbing paper strip will be placed inside the gingival sulcus until it feels the resistance. One of the main disadvantages in this procedure is that this paper can itself induce various irritations into the saliva, which itself will produce more excess of the gingival crevicular fluid. So, in order to overcome this disadvantage, they developed the extraclavicular paper strip method. In this method, the absorbing paper strip will be placed at the opening of the gingival sulcus so that the fluid that seeps out of the gingival sulcus will be collected by the paper strips, as you can see in the image. Now coming on to the twisted threads. So before going on to these twisted threads, the area should be isolated. Then the twisted threads will be weighed before placing into the gingival sulcus. After placement into the gingival sulcus, again these twisted threads will be weighed. So the difference in weight gives the quantity of the GCF collected in the twisted threads. Now let us see about the micropipette. This micropipette acts by the capillary action. Once the fluid is collected, later these fluids can be centrifuged and analyzed for its contents. The last method is nothing but the intraclavicular washings. Previously, the device consisted of a device which is made up of the acrylic plate, which is placed over the maxilla with soft borders running over the gingival margin. Along these soft borders, there will be grooves. In the grooves, the fluids will be collected into the 4 collection tubes. But recently, we are using just 2 needles. As you can see in the image, the first needle is the saline flush. You can see the tip of the needle at the bottom of the gingival sulcus. The second needle is for collecting the gingival crevicular fluid. The collection is through continuous suction. Now, coming on to the amount of GCS and how it can be detected. In the paper strip, we are collecting the gingival crevicular fluid. Either we can wet this paper using the ninhydrin stain. After staining, these can be viewed as a larger photograph or under the microscope, or else it can be used to detect via the electronic transduction. The wetness of the paper, usually it will interrupt the electric signals that are passing through, which will give out as the digital readout. Now coming on to the composition, as said earlier, there are various cells seen in the gingival crevicular fluid. Few among them are the bacterial cells, our host cells, which consists of the leukocytes, which may also present as neutrophils, lymphocytes, macrophages, or the monocytes. Now coming on to the electrocytes, various electrolytes are present in the gingival clavicular fluid. Few among them are the potassium, sodium and calcium. Now coming on to the organic components, among the organic components, carbohydrates, proteins, the metabolic products and the bacterial products are seen in the gingival clavicular fluid. Among the carbohydrates, the most commonly seen are the glucose, hexosamine and the hexuronic acid. The glucose content in GCF is always 3 to 4 times higher than that found in the blood products. Whereas now coming on to the proteins, in contrast to the carbohydrate, proteins are found in lesser quantity in the GCF when compared to that of the serum. In the metabolic and the bacterial products, various bacterial products are found as well as the metabolic products are found in the GCF, which includes lactic acid, urea, hydroxyproline, endotoxin, cytosolic substances, etc. Now coming on to the clinical significance, why this GCF is important. This GCF plays a very important role in diagnosis and also as well as the management of the diseased tissues. Its presence in the normal clinical gingiva, it suggests that even though the gingiva is clinically healthy, when we view these gingiva in the microscopical sections, there appears some inflammation. Usually, this GCF is not increased by trauma from occlusion, whereas vigorous brushing, the ovulation during the pregnancy time or other mechanical factors might increase this GCF production. Now coming on to the clinical significances. This GCF quantity, it will increase from 6 am to 10 pm and thereafter it decreases. This difference is known as the circadian periodicity. As said earlier, during the trauma from occlusion, this GCF is not going to get increased, whereas during the mechanical stimulation like massaging the gingiva or during tooth brushing, this GCF fluid is going to increase in quantity. Now coming on to the sex hormones, these female sex hormones, they are going to increase the gingival blood supply, thereby increasing the GCF production. During the smoking, initially there will be increase in the gingival clavicular fluid flow, but as the time progresses, there will be a decrease in the GCF flow. Whereas after the periodontal therapy, there will always be increase in the gingival clavicular fluid during the healing phase. Now let us see what are all the drugs that can be excreted in the GCF. The most important drugs that can be seen in the GCF were metronidazole and the tetracycline. Now coming on to the second part, that is the leukocytes. These leukocytes, they can be found even in the clinically healthy gingiva under the base of the pocket in the connective tissues, even though there are no inflammation seen in the adjacent connective tissues. They travel across the epithelium to the gingival sulcus and are expelled there. These leukocytes appear extravascularly in smaller number in connective tissue near the apical area of the sulcus. Among the leukocytes, the most common leukocyte you see here is the PMNS, that is 91.2% are PMNS and 8.5 to 8.8% are mononuclear cells. The other mononuclear cells are composed of the 58% B lymphocyte, whereas only 24% is T lymphocyte. This is typical because in the blood you can see there is a more amount of T lymphocytes when compared to B lymphocyte, but in case of the GCF, the T lymphocyte to B lymphocyte is reversed from that of the blood products. Now these leukocytes, they are attracted by different plaque bacteria but are also found in the dentogingival region of the germ-free adult. Most leukocytes are viable with phagocytic and killing capacity. We all know that phagocytic is nothing but engulfing of the bacteria by the leukocyte. Also, there are live and dead leukocytes that can be found in the saliva, with the main entry point of the leukocyte into the oral cavity being the gingival sulcus. In this image, you can see there are holes. Through these holes, the leukocytes will be emerging. You can see the leukocytes emerging from the holes here, and you can see small radio opaque area which is the bacteria. In this image, you can see clearly this is the leukocyte covered by numerous amount of bacteria, and this crossed arrow you can see the leukocyte engulfing the bacteria, and this process is known as the phagocytosis. Now coming on to the saliva. As we all know, saliva has various functions, like it mechanically cleanses the oral cavity, it can buffer the acid that are produced by the bacteria, and it also acts against the bacteria through the immune mediators. Saliva is considered a main biologic fluid for various potential biomarkers similar to the gingival clavicular fluid. Now coming on to role of saliva in the oral health. As we can see, these glycoproteins and mucoids, they are nothing but coating agents similar to the gastric mucin. This helps in both lubrication and the physical protection property of the saliva. Next, coming to the cleansing property, the saliva through its mechanical property, that is the physical flow, it cleanses all the debris and bacteria from the teeth surfaces. That is the cleansing property. Now coming under the buffer, the acid produced by the bacteria are buffered by few enzymes like the bicarbonate and the phosphate. Now the minerals present in the saliva that maintains the tooth integrity. The last comes the antibacterial action, that is the immunoglobulin A and other enzymes like the lysozyme and the lactoperoxidase. This provides the antibacterial action. Now coming on to the antibacterial action in detail. As we said earlier, the inorganic factors, few examples can be bicarbonate, sodium, potassium, phosphate, calcium, fluorides, ammonium and carbon dioxide. Now coming on to the organic factors, it consists of various enzymes also, lysozyme, lactoferrin, lactoperoxidase, defensin, peptides, etc. Now coming to detail, the lysozyme, this is one of the very important enzyme. This enzyme works on both the gram-negative and positive organisms. It also repels the transient bacterial invaders. The main mechanism behind this action is that it cleaves the linkage between the structural component of the glycoprotein and the muramic acid. This is the main mechanism that acts as the antibacterial property. Now the second one is the lactoperoxidase thiocyanate system. This is bactericidal especially to some strains of lactobacillus and the streptococcus. Also there is one more enzyme, that is the lactoferrin, which is effective against the lactobacillus, that is the actinobacillus species. Now the myeloperoxidase, it is usually released by the leukocyte. It is bactericidal for actinobacillus, but it also has the additive effect of inhibiting the attachment of the actinobacillus strain to the hydroxyapatite. Now coming on to the antibodies, there are various antibodies that are found in the GCF, also in saliva, like the IgG, A and M. Among these antibodies, the IgG-A is the main antibody that is found in the saliva. This IgA is produced by major and minor salivary glands, whereas the other two antibodies, that is the IgG and M, are introduced into the saliva through the gingival clavicular fluid. Now coming on to the enzymes, the sources of the salivary enzymes can be various. We can see what are all the sources of the enzymes. One is the salivary gland, bacteria, leukocytes, oral tissues and the ingested substances. Now let us see what is the major enzyme that can be seen in the saliva. The major enzyme is the peroxide amylase. Now let us see which enzymes can be increased in the periodontal disease conditions. These enzymes includes hyaluronidase, lipase, beta-glucuronidase, chondroitin sulfatase, alkaline phosphatase, catalase, peroxidase, collagenase. These are the few enzymes for example. Now coming on to the buffering capacity and the coagulation factors. In saliva, the most important buffering mechanism is nothing but the bicarbonate carbonic acid system. Saliva also contains various coagulation factors. The buffering action is mainly by maintaining the hydrogen ion concentration at the mucosal epithelial cell surface and as well as the tooth surface. Now among the coagulation factors, the most common factors found are the factor 8, 9 and 10. In addition to these coagulation factors, also active fibrinolytic enzymes can be present in the saliva. Now similar to the leukocyte, as we seen earlier, the leukocytes are also found in the saliva. There can be live leukocytes, also dead leukocyte. The leukocyte, especially neutrophils found in the saliva, are known as the orogranulocytes, and the migratory rate is known as the orogranulocyte migratory rate that are found in the saliva. Now coming on to the characteristics of these PMNs that can be found in the saliva. The whole blood PMNs are naive and are non-activated. The PMNs in saliva interact with multiple antigens from the microbiome. This can also increase in the presence of the gingivitis. The PMNs reach the oral cavity by migrating through the cellular epithelium. Now coming on to the role of the saliva in the periodontal pathology. We also know that saliva is helping in the initiation of the plaque and its maturation. Not only this property but also the composition and the flow of the saliva can influence the calculus formation as well as the periodontal disease progression. Now in addition to this, if there is a decrease in the salivary flow, it can result in various diseases including dental caries, xerostomia it is the condition where there is a decrease in the salivary flow. It can also result in the periodontal disease progression. Now coming on to the periodontal therapy, immediately after the periodontal therapy, there is going to be increase in the salivary flow. Now we have seen all the defense mechanism of the gingiva. So in conclusion, I am going to conclude that the defense mechanism of gingiva is multifaceted and highly effective in protecting the periodontium from the bacterial invasion. Thank you. I thank you everyone for watching this video.